Roberto for this. Hi everyone, thank you very much, Roberto, for this kind introduction. Very glad to be here today and tell you all about my research on copepods and how they counter dispersion to maintain high encounter rates in turbulent flows, or basically dispersion. As Roberto said, I'm primarily come from the turbulence community. And then I did my postdoc back in Zurich, studying copepods and basically how they disperse around in their environment. And these are some of the results that we have obtained. So this is basically my background. I come from fluid mechanics. I studied turbulent flows, dispersion of scalars of particles, viscoelastic flows a little bit, and this is now the copepods part of my diverse research. I usually, like my origin from the PhD came from actually studying Lagrangian dynamics in turbulence. So basically tracking particles in 3D in different turbulent flow configurations. And then we thought that we can implement these experimental techniques that we have for tracking stuff in 3D in order to see how plankton, and in particular copepods, behave in turbulence. So this is kind of my connection to this field through this particle tracking connection and through dispersion, which I did before for at least for Lagrangian particles in fluid mechanics. Now I did it for copepods. So before diving into the topic, I want to briefly say thanks to my collaborators. So we have Professor Markus Holzner from Zurich, who was my PI when I did this work. We have François Guérin-Michalek, who was kind of my predecessor in the group. He worked a lot with copepods in Marcus's group, and I kind of inherited some of my research from him. Some of the topics involved from François. And then we have also Thomas Kiobo, who helped us a lot with working with live specimens of copepods, because as I said, I was not an expert really on breeding copepods, on obtaining the samples and everything. So having Thomas Kiobo to help us with that was an amazing thing and a very important collaboration that we had. So copepods are basically planktonic crustaceans. They are, in fact, the most abundant animal group in the ocean, apparently. There are many different kinds of copepods. Some of them are seen here. This is an image taken from Wikipedia. We can see that there are a very large variety of them. And in particular, this, for example, species, this is Temora longicornis. This is the particular species of copepod that I was working with in the lab. So this is an image of a specimen that we used in our lab. Copepods have a really important role in the marine food chain. So as you all know, phytoplankton basically feeds from sun, from the light. This brings up the primary production in the ocean. Then these are being fed by zooplankton, among which we have the copepods, which are then being fed by larger predators. And also their feces contribute a big part of the global carbon pump process. So they really have a very significant role in marine ecology. So coming from the etymological point of view, plankton comes from Greek, and it means basically drifter. So when people first thought about plankton, they thought about passive drifters in the ocean. However, thinking of, at least of copepods as being entirely passive is not really true, because plankton and copepods, in fact, can swim, as can be seen right here. They swim in two basically different modes. They have this slow kind of drifting mode of swimming. And then they also have this jumping mode of swimming. So basically, they're not so passive as people used to think in the past. They swim for various reasons. So for example, they swim in order to escape predators. In order to escape from predators, they basically jump. These jumps that we saw are very quick, and they can help copepods to really escape from the suction feeding current of a larger predator. They also swim in order to find and eat food. So they need to basically search their environment and then swim through the water column in order to filter feed of the phytoplankton in the water. And then they also swim in order to find mating partners. So at least the species that we were working with, and a lot of other species, there are males and females. And in order to reproduce, males and females have to encounter one another and interact sexually. This means that they need to search for one another, to pursue for one another, meaning that this has an important aspect of their life. And here we see basically overlaying quite a lot of images of copepods swimming in my tank at the ETH Zurich. We can see they're fairly active, I should say. Because they swim, they basically disperse in their environment, right? So they swim, they don't really, you can say they're roughly blind. They do have some photoreception, but it's very limited, meaning that they would essentially swim randomly in their environment. And this causes them to basically diffuse in their environment. A second reason for why they are diffusive is because of the turbulence in which they live. So if we have a random turbulent current in the water where they happen to reside, they would just be drifted by the current randomly, essentially giving them a diffusive kind of nature for their motion in the ocean. And this has quite a severe consequence for them. In particular, if we take a group of copepods that is initially very close to one another, and we just set them free in the ocean, then due to these two reasons, we expect them to, over time, be dispersed away from each other. So this initial small cluster of copepods is going to end up as a very big cluster of copepods where they might not be able to interact anymore. And this is quite problematic from the mate searching perspective, because if it turns out that if we had assumed that the copepods over the oceans are uniformly distributed and that they swim randomly, such as Brownian particles, then the random rate of encounters that they would undergo would be insufficient in order to support their own population, right? So this means that copepods cannot, at least with the number that we have in the fields, counted by people in field experiments, we cannot have copepods uniformly distributed all over the ocean. This means that this scenario that I just described of groups of particles of copepods being separated away from each other is quite problematic. And in order to counter this effect of basically separating away, copepods are found to live in small groups. These groups are called mating clusters. These are clusters about 10 meters in size, a few tens of meters in size. These have been observed in the ocean, and this is kind of how copepods keep together. So on the one hand, copepods swim around and they're being drifted and dispersed by the currents around them. But on the other hand, they must be able to encounter one another in order to reproduce. And they must be able to maintain these mating clusters. So their life is kind of a trade-off between searching for food and searching for mates while maintaining high encounter rates. So the question that we asked in our study is how can copepods maintain high encounter rates and keep their patchy distributions when they're constantly being diffused away from each other. So this is the question that we wanted to kind of tackle in this research. In addition to that, copepods can interact with each other. Apparently, copepods have two basic means of communication. The first one is hydromechanical sensing. So if we have two copepods swimming one by the other, then the disturbances that one of them leaves in the flow behind them in their wake can be sensed by this very sensitive antennas of the other copepods. So this guy can sense the disturbances that this guy senses, and then this guy can guess that there's another copepod in his or her environment. The second means of communication is through pheromones. So apparently, female copepods leave a trail of pheromones as they swim in the water. And male copepods have chemoreceptors that can sense this pheromone trail and then trace it up in order to reach these females. So this is how the males can find females. As a result, male copepods need to constantly search their environment for cues from nearby individuals, either these hydromechanical cues or these pheromone cues. And then when they do pick up a signal, they will try and attempt to make contact in order to verify whether mating with this other individual is possible. So for example, let's see this video right here. Here we have two copepods. And this is kind of the picture that I'm trying to convey. We have one copepod right here. He's starting his trajectory, he's swimming away, and then he senses that there's some kind of other disturbance right here. So then he turns back in order to see, I mean, what was that I felt? Obviously, this is my interpretation, but this loop that this guy was going through, I mean, I can't really see any other reason why this guy went back to its original position. And now my whole talk is going to be based on this kind of a mechanism. I'm going to say, or I'm going to propose the idea that this approach for contact that copepods have for one another can lead to clustering of individuals and might lead to the clustering that we see in the ocean. Okay. So we have mating clusters. This is an experimental fact, but how? And we'll present a brief theoretical framework and an experimental verification for this theoretical framework that we have developed. So in order to test this idea, we basically built a stochastic model. It's a very simple stochastic model. It's not meant to reproduce all of the aspects that the copepods have in their life, but only to try and kind of reproduce this clustering type of effect that we saw. So these are the following rules that particles or copepods undergo in our stochastic model. First of all, copepods are represented as basically points in space that move randomly. Each of them is kind of like a Brownian particle just moving in 3D space. Then each and every one of these particles is associated with an interaction sphere, right? There's a region around each and every one of these particles that we call this interaction radius, okay? So if we have two copepods in our model that are at one particular time step inside of each other's interaction sphere, this means that they have detected one another, and that in the next time step, seen right here, they will form an interaction. Basically, this interaction is being modeled as both of these particles moving to the same location for one time step. And then in the following few time steps, this is a parameter of the model, the number of time steps during which particles can no longer interact with one another. This is a fixed amount of time steps that we denote as M. We call it kind of a memory timescale during which they remember that they had an interaction. And during this memory timescale, they cannot interact anymore with any other particle. Okay, so this kind of prevents basically particles just sticking together indefinitely after a single interaction, this memory timescale. And this is it. This is everything that our model has. We can represent this behavior using this equation. It's kind of a long equation, but it's actually very simple. So the position of each one of our particles or model copepods at time I plus 1 is equal to the position at the previous time step, which is basically xi, plus a random direction vector. This is theta, that we choose it randomly in every new time step that we go through, times a fixed step size that we have in our model, plus this big old term that accounts for the interactions. So we have basically two indices. The first index is a distance index. This counts whether they do have one particle within their interaction sphere or not. And then we have the second index right here that accounts for this memory timescale process. And then if both of them are true, are equal to 1, namely they did not have an interaction in the previous n steps, and they find themselves in a close proximity to another particle, then both of them will basically just be moved in the center position that both of them had in the previous time step. So it's a really very simple model in which copepods are modeled as random walkers that have attracting close range interactions that can turn off and on based on their memory and on the distance that they have with respect to other particles. Okay. The model has two main effects. The first one is that we have a reduced diffusivity of particles that have interactions. And this can be very easily seen by these particles that have been integrated numerically in our model without any interactions. We can see that this is the area that they occupy after 500 time steps, versus these groups of particles that do have interactions, and after the same amount of time, they cover a significantly smaller area. This is basically can lead us to understand that they have a reduced diffusivity. Once we turn on interactions, particles tend to spread out. They still tend to spread out, but much slower as compared to the no interactions case. So we have a kind of an anomalous diffusion that happens in our model, and I'll go into that very shortly. And then the second mechanism that we can see is small-scale clustering, what we call small-scale clustering. So if we compare this distribution that we have, for example, with this distribution that we have, we can see that almost all of the particles in the right-hand side, for the particles that did interact, almost all of them have at least one nearby neighbor, right? So here, here, here. Some of them do not. In fact, only two of them in this case don't have nearby neighbors, this guy and this guy. All of the rest of them do have them. We call this this observation small-scale clustering. And again, I'll go into a bit more into details about small-scale clustering shortly. So just to describe this anomalous diffusion property that I mentioned, if we have Brownian particles, then their mean squared displacement of these particles is going to be equal to 2 times d times the time of diffusion. This is an analytical result for Brownian diffusing particles. If they're a simple Brownian particle, this d, the diffusivity is going to be constant. However, if this d coefficient is a function of time, then we call this an anomalous diffusion case, right? So for normal diffusion, d is constant. For anomalous diffusion, people tend to specify it as a power law can specify it as power law of time. So if we have this d changing with time, we have an anomalous diffusion case when alpha is different from 0. Yeah, and what we can see here is that initially, at very short time scales, the diffusivity, actually here in this left-hand side of the plot, we see that we plot the diffusivity normalized by the initial value of the diffusivity at time equals 0 for one of the rounds of the model. We can see that the diffusivity definitely changes as a function of time in our model. So, and in fact, it decreases as a function of time. This means that we have a sub-diffusive process at small time scales. Okay. This this subdiffusion process that we have, we associate that basically due to the interaction of the particle, one with another. At longer times, we can see that diffusivity stops to decrease, at least asymptotically, and tends toward a constant value where we have a normal diffusion at longer time scales. This is because at longer time scales, particles can basically jump from one cluster to another. And this leads to a long time, kind of a normal diffusion case process at longer times. So this is our interpretation of the results for the mean square displacements of the particles in our model. We should say that even at long time, the diffusivity is still lower for interacting particles versus non-interacting particles. And this lower diffusion, right, the final diffusivity at very long time is equal, at least here, only to half of the initial value, right? And this is what essentially leads to this small rate of expansion of the whole group of copepods that we saw in the previous slide. Now let's talk about small-scale clustering. In order to analyze this quantitatively, we can do the following kind of analysis. We take the whole domain, the whole numerical domain that we have in our model, and then we divide it into smaller beans. These beans have two smaller cubes. These small cubes have a side length of L, and they're basically cubes, symmetrical cubes. And then we can go about all of these beans or these cells and count how many particles we have in each and every one of these cells, right? So for example, this particular bean has 3 particles. And we denote this number of particles as being K. So here, K is equal to 3. For this cell, K would equal to 5. Apparently, it can be shown analytically that if particles were distributed homogeneously across all of our space, what we would have obtained for the PDF of K of L is simply a Poisson PDF. This is an analytical result, a very nice result that people know. This would correspond to this black line right here. However, as it turns out, if we do the same kind of counting of number of particles in each cell for the particles in our model, we get a completely different result, as can be seen right here in the blue dots. This is the result of one of our run of the model, and we can see that for small number of K, the value of the PDF is slightly lower than the Poisson distribution, meaning that we have less beans that have only 1 particle inside of them. However, we have a lot of significantly much more beans that have higher value of K, right? So we have many more such beans that have very high values of very high numbers of particles inside of them. You can say, okay, well, this PDF goes down to very small numbers, right? It goes down to 10 to the minus 4, 10 to the minus 6, 10 to the minus 8. So is this even significant? And I would say, I think it is significant, because as can be seen, for example, here in this inset, when we plot this PDF normalized by the Poisson distribution in linear log scales, the relative the ratio between these two PDFs grows exponentially with k. This means that if the sample that we have are large, then the effects that we have as k increases become more and more and more significant. So I think it's quite a significant effect eventually. So let me just make it clear. For example, here we can see here in this inset that that for k equal 12, for example, the probability was higher by a factor of 200 as compared to the uniform distribution. So it's quite significant, a higher probability by a ratio of 200. So clustering, but why? I mean, I think I tried to convince you that we do have clustering in our model. Also, using this kind of plot, this was done in 2D in order to have a more strikingly visual effect that is more prominent in 2-dimensional simulations as compared to 3-dimensional simulation, but still. So in order to kind of try and explain why do we have this clustering, then as we saw before, particles have a reduced diffusivity because of these interactions, meaning that when we have a few particles nearby one another, they tend to interact with one another, which reduces their diffusivity, their self-diffusion. And if these interactions reduce their self-diffusion, it means that they travel smaller distances, thus increasing the probability to have large clusters. Basically, particles tend to agglomerate where other particles are due to the interactions that they have with their neighbors. This is kind of a heuristic description of this probably more complex picture, but this is what we think is going on in this model. So now let's try to confirm this model with experiments. In order to do that, we basically did this 3D particle tracking experiment that I described to you before. This is a technique that I've learned, at least, from the turbulence community. There, we like to track particles in 3-dimensional space. Here, we simply tracked copepods in 3-dimensional space. We have 2 different experiments. One was performed by me using one species of copepods. I used Temoa longicornis. The second experiment was done by my predecessor, François Michalet, at the same lab, but he used a different species of copepods. He used Temoa affinis. Overall, each and every one of our experiments has about 10 million data points. And at least in my experiment, I recorded data for about 15 minutes of data. Another difference between these 2 experiments is that we used very different values of the density, the number density of the particles inside of the tank. So François, with his E affinis experiment, used a very high density of copepods, about 2.7 on average. 2.7 copepods per cubic centimeter. However, I use a very low density of copepods inside of my tank. With the Temoa longicornis, I had 2 orders of magnitude less value in the density. So we have really 2 completely different experiments. We use the same technique. We had this, I mean, it was the same lab, but there are 2 different species, and there are 2 very, very different densities of specimens inside of the water tank. So in order to try and see if our experiments and the model correspond to one another, we have to measure the parameters of the model using the result of the experiment. The model basically has two important parameters. The first one is the interaction radius. So we need to be able to measure the interaction radius independently of the clustering probability in order to confirm our model. In order to do that, what we do is we look at the relative velocity that the two copepods have between them. So if we have two copepods swimming one by another, they have a distance between them, r, and then we can look at the RDT, the rate of change of the distance between them. This would correspond to the separation velocity of the two copepods. If it is negative, it means that the two copepods are going towards each other. If it is positive, it means that they are going away from each other, right? And then we can plot the average value of the separation velocity as a function of the distance between the two particles. What we saw, and this is a technique that was invented by Francois, he saw that males and females, when we plot this function, has two very different behaviors for males and for females. Apparently for females, shown in red, the average value of the separation velocity is roughly 0. It doesn't change really significantly as we change the distance between the particles, although here maybe we have a higher positive values, meaning that two females, when they're very close to each other, they want to go away from each other. They don't really like to be near other individuals. That's for females. And then for males, we see a completely different behavior, where at large distances, where presumably the copepods cannot feel one another, the average value of the separation velocity is roughly 0. But then as we look at smaller values, we see a very strong signal of this separation velocity becoming negative, meaning that males do like, when they see another thing next to them, to approach it. This allows us to define the interaction radius as the value of r in which this law, this average separation velocity, starts to become significantly negative. So we can measure this A using this graph, this interaction radius using this kind of graphs. We did that for the Eurotomoa finis, and we got about 4 millimeters. This is the interaction radius of Francois' copepods. For my copepods, we got a very similar value of about 5.5 millimeters. Slightly larger, but roughly on the same order of magnitude. I should say this is approximately about 2 body lengths of copepod, of the copepod that we use. The second parameter that we have is the memory timescale, this M. We need to be able to determine what is the memory timescale in order to fit it into the model without relying on the clustering statistics that we have in the experiment, in order to have it like a really independent test. So in order to do that, we looked at the distance that each and every one of the copepods we had in the tank to its own nearest neighbor, right? So we have one particle going about, swimming in the tank, and we always measure the distance between this guy and his own nearest neighbor. And then we plot this nearest neighbor distance as a function of time. Right here, this line, for example, corresponds to one particular copepod that I tracked in my experiment. This was a track of 8 seconds long. We can see that there are two basically critical values. The first one, I mean, since we know what is at least presumably, we know what is the interaction radius between the copepods, then we know that if this guy, the copepod corresponding to this line, is smaller than the interaction radius, it means that it was probably able to feel its nearest neighbor around him or her. And then the second critical distance is touching. So if the distance between this copepod and its nearest neighbor is smaller than their own body size, taken as, I think here it's 2 millimeters, then we consider them to be touching one another. So this is like really an interaction, a physical interaction, not just a sensing kind of interaction. And then in order to estimate what is the memory time scale, we can look at the durations of time, right, that have passed between interaction events. So if the copepod that corresponds to this line wasn't started an interaction at this time instant, then it was interacting, then no interaction for some period of time, and then poop, starting another interaction. If we look at the time between these two consecutive interactions, then the statistics of this might be interpreted as the memory timescale that we have for the copepod in our tank. Again, this is an estimation, but it's the method that we have introduced. So basically what we did is we calculated these kind of graphs for all of the copepods in our tanks. We looked at the consecutive interaction events, measuring this tau, the time interval that occurs between the interaction events. We plotted the distribution of tau, and then we took the median value of tau, of the distribution of tau, to be the memory timescale in our experiment. So this is kind of a statistical estimate based on the observations of these nearest neighbor distances. We did these calculations for the Eulogimola finis, for Francois' database. We got about 2 seconds for the memory timescale. This should be A, this should be N, sorry for that. And then we did the same kind of calculation for the Temor longicornis data set that I gathered, and we got about double the value, almost 3.7 seconds, almost double the value. Right, so this allows us to kind of estimate what would be the memory timescale. Of course, we're relying on the assumption that copepods can always interact with someone in their environment. We assume this to be true. That's an assumption. Maybe its accuracy can be checked, but this is an assumption that we rely on when we calculate the memory, when we estimate the memory timescale using this method. The thing is that now that we have an estimation for the interaction sphere and for the memory timescale, we can actually see and test whether our model can predict the observations for the small-scale clustering effects that we have in the experiment. So right here, we have in black line the Poisson distribution that corresponds to the same density that we had in the Eulogimola finis experiment. And here we have the Poisson distribution for the Temor longicornis experiment. Both of them look, I don't know, they're here, shown on continuous lines. And then for the experiment, we just measured the small scale, this binning, this K of L PDF, also in our experiments. And we have obtained the PDFs shown right here in these orange squares. We can see that, at least for the Temor affinity, is significantly higher. For the Temor longicornis, it is still higher at higher K values, although the resolution of our measurement could be improved. I do admit that. And then let's see what our model did in comparison to the results of the experiment. This can be shown right here in the blue circles. And I mean, I think the graphs kind of speak for themselves. The result that we got from the model as compared to the experiment was quite good, I would say. And this is using the memory timescale and the interaction spheres that we have estimated from the direct empirical data. So this, I think, gives us quite a good indication that something in our model, at least, is in the right direction for predicting these small-scale clustering effects. So to conclude this talk, we have this kind of scenario where copepods, if they begin at small-scale clusters, we would expect them to be dispersed away over time due to turbulence in their environment and due to their own swimming. However, they can somehow maintain small-scale clustering, such as these kind of effects. And we think that what causes this small-scale clustering effect is due to these repeated interaction mechanism, this high five kind of effect that I've described in the model. So when copepods cross one another, they tend to come back and to, at least males, they tend to come back and to try and sense, can I mate with this other individual? If they can, great. If not, they don't. But nevertheless, this repeated movement, this coming back towards the other individual that they saw, can support small-scale clustering. So just to read what I wrote, our work shows how copepods balance between searching for mates and for food while remaining gathered in groups. Small-scale clustering results from a unique mate-seeking behavior of copepods. I mean, these repeated interactions, as we saw in the movie, and we get, I think, quite a good quantitative agreement between the model and two completely independent laboratory experiments. We hope this suggests, at least, I think, that our approach can be leveraged in order to predict clustering, mating clustering, and encounter rates of planktons in actual marine environments, which is quite a crucial step in order to be able to model copepods reproduction rates. And with that, I thank you very much for your attention. I will leave you with this movie of repeated interactions between these two copepods, basically dancing around one another. So thank you very much for your attention. If there are questions, I'd be happy to answer.
Thank you very much, Ron, for this very nice talk. Now it's time for questions from the audience. If anybody has a question, you can just unmute yourself and ask the question.
I have a question.
Yeah.
This is Jeanette Yen over in Georgia Tech. I was wondering, I know your particles don't have any behavior. Is that correct?
In the model? Yeah, definitely. They're just random walkers. They go about wherever they go. I mean, it's completely a random process.
Well, I observed something really cool this summer. In my copepod Yukita, the males swim up and down and the females swim in 3D.
Wow.
It was amazing. So I would like, in my mind, it sounds right to improve encounter rates for one animal to swim up and down and the other animal to be kind of.
I'm sorry, Jeanette, I for some reason I can't hear you.
Oh, can you hear me now?
Yeah, now I can hear you again.
Oh, okay. I think I have to.
Sorry again, it's a bit difficult. There might be some connection
near the speaker at the base of the phone.
Okay.
Okay. Well, what to say then?
I'm sorry, I really, for some reason, the connection was not perfect. Could you please repeat the comment? I'd be very happy to hear your comment.
Oh, first you have to let me, if you can hear me. Can you
now
hear
I can
me?
hear you.
Yeah.
And you couldn't hear me before? Did we change something?
I don't know why, for some reason it got stuck. Maybe I'll stop the sharing to improve the connection, then we can speak better.
Oh, okay. I'll stop the video for me. I'll stop the video and just hope that that might do it. But anyway, I think most people got the gist of it, that in this one species, the males and female.
I'm sorry, Janet. Again, I really apologize. For some reason, something happened to the line, and
Maybe if you have
somebody
a
else
question,
can-
we can type it in the chat.
yeah, that could be a great idea.
Sophie, maybe you had a question. Maybe you want to go on, and then while Janet is typing in the question.
Yeah, I just had a quick question. Super cool talk. Thank you. That observance, the experimental observance of the different behavior between the male and the females was super cool. But I was just wondering, was that always a mixed population? So was it in response to the pheromones, or if you did all male, would you still see that response?
So in order to really distinguish the females from the males, we have to do a separate experiment for only males in the tank, and then another experiment for only females in the tank. Because as you saw, our videos are taken from a long distance in order to have a full view of the tank. So we don't have a sufficiently high resolution in order to determine whether the individual that we're looking at is a female or a male.
So the difference in male response is not due to pheromones or anything. That's just they naturally respond differently to stimuli than female. Okay.
Yeah, I also tend to think that in our tank, because it's not a very big tank where we do the experiments. It's about this size, only 10 centimeters by 10 centimeters.
Mhm.
I think it would be mostly dominated by this hydromechanical type of interactions, because if the females constantly release pheromone into the water, eventually the water will be saturated. At least in my mind, the water might become saturated from the pheromone, and it might be difficult for the males to actually distinguish that from the background concentration. Maybe I'm wrong, but that's what I think.
Awesome. Thank you.
Thanks.
Ron, thanks a lot. Cool talk. I have several questions. So what is natural density of copepods in nature?
So it really strongly fluctuates. That's the thing. This clustering that I was telling you about in the ocean, and I'm sure maybe there are people who know more about that in the participants. Probably Jen knows more than I do. When people actually look at oceanic measurements of the copepod concentration, they fluctuate really strongly. It's a highly intermittent kind of a signal when people look at that. Sometimes people see 500 individuals per cubic meter. Sometimes people can see one individual or even 0 individuals per cubic meter. And this is a really intermittent kind of signal. So I would say the densities that were in Francois' experiment are a bit higher than what people have in ocean measurements. But what I used, which was 10 orders of magnitude lower, would be closer to what we have in the field.
I see.
Yeah.
And how would you choose this L parameter, the size of the tile that you used for the statistics of the Poisson versus non-Poisson?
So I checked. Obviously, it's used by you can set it yourself. I mean, it's not based on the model, it's only for the interpretation of the results. And I checked for various values of L that we get at least consistent results. I mean, the Poisson distribution itself would depend strongly on L, but then it would still be lower as compared to the distribution you get from the experiment. The important thing to do is to match the density, the number of individuals, the average number of the individuals per cell. And this is the important parameter of the Poisson distribution.
And I also was wondering, so in the life cycle of copepods, do we know anything about when they mate? Like, do they like typically mate, or for this food versus mating trade-off that you mentioned?
Mhm.
Do we have
Well,
any idea how much time they spend doing one thing over the other?
I cannot really tell you like what
Okay.
is the fraction of time that they would search
Mhm.
for mates versus for food. I can say that they live, at least the species that I used, live for around 3 months.
Mhm.
Their juvenile stage lasts about 1 month, and then the males start searching for females.
I see.
I can say from the experiment that we had, because actually it was very great that Jeanette was here, because they were the first ones to actually do this pheromone measurements of
Mhm.
copepods, and we mimicked them. Actually, we mimicked Jen's results. You basically take water that are saturated in pheromone, and then you inject it into a tank that has only males inside of that, and you can actually see the response of the males, like really shooting up the pheromone trail, and it's quite an impressive thing, because you see them basically swimming around very slowly, and then you really see them like catching up something, so they start like moving fast, and then swim up very fast while the pheromone trail is going down. So that's a very nice thing. Yeah, I can't really
Cool.
say. Yeah,
It's
it was.
good your species does it too.
Yeah, I was really fascinated to see that. Really, it was amazing.
It's amazing. It's really, truly is. And what I saw for this Yukita, because this is only the beginning of me trying to get them to mate, because all the other species were too big. I couldn't give them enough room. So, but I did separate the males from females. So the young woman who asked, I did do that, even though it was really hard, because it was like female to male was like 99 to 1. So it took me like 2 hours just to get to 20 males.
Wow.
But I saw them spin. And that's what Temera does. When Temera finds a trail, he spins to relocate the trail. And that thins the boundary layer near their heathostasks so that it doesn't have to diffuse so far. So it's a very complex but fascinating behavior. I'm so glad you saw it.
Yeah, I was really happy when I saw it in my videos. Really, I have some
Yeah.
recordings of that. I could search it in my computer. It was really amazing.
Very good.
More questions? Okay.
Okay, I still need that to be clarified. So when you saw the males swimming funny, was that males with pheromone in your water?
So I took a bunch of males, let's say 10
Yeah.
males, and I put them inside of the tank in
Yeah.
the dark, and I left them in this tank for, let's say, 10 hours, something like that. And then I took water from a separate tank that had only females,
Yeah.
and I injected it. Well, it was slightly heavier water because I put just a slight bit higher concentration of salt in order to get a
Yeah.
really fine
Very
trail of this
nice.
thing. We injected it using a syringe pump at a very low rate. So
Yeah.
it really creates a like kind of a column of the female water inside of the middle of the tank. And then we had a few of the males like sitting on the bottom of the tank. And then 2 of them, they started to be agitated. And then you saw one of them really jumping up, at this very high velocity that they do, just
Yeah.
reaching up to the tip of the needle. and then sinking back down. Then he caught the
Wonderful.
trail again, went up, and then went
Wow.
down again. And then it repeated several times over the course of like 50 minutes, something like that.
But those
Sorry, I couldn't hear again.
weren't the data you used in your presentation to compare to the rand. Okay, so you
No,
used just pure no scented water. Okay, good. That's good. Then I get
Yeah.
it. Yeah,
Yeah,
no,
yeah.
the scented water does something funny to them. Okay, I get it now. Thank you.
Thank you for this comment.
Yeah, thank you.
More questions? Okay, there seem to be no more questions, so I think we can thank again Ron for this very nice talk.
Thank you. Thank you for having me.
Thank you.
Thank you.
Thank you. And we'll send out again an email later on for July's talk as usual.
And we'll put the recording on the YouTube channel.
Great.
Okay, thank you.
Thank you.
Thanks,
Bye-bye.
guys.
Thanks,
Bye, bye, bye.
evening, everyone.
See you. Thank you, Ron.